Hello, this is my presentation on the study that I did on the gut microbiota composition in fruit flies when comparing presence of live yeast to no presence of live yeast and comparing male to female flies. So the objectives for this study were first to analyze the effects of the fly sex has on microbiota composition, and second to analyze the effects that live yeasts have on microbiota composition in the fly. So the first objective came to be because female flies have a larger body size than male flies do, and as such, they could house more bacteria, and more bacteria could mean that there's a change in the composition of the microbiota. So we wanted to see if that was an effect that happened. The second objective came across because in the wild, fruit flies have they eat live yeast, and these live yeasts make an important part of their microbiota. However, in the lab, we only feed them dead yeasts, so I wanted to see if live yeasts had an effect on the composition of the microbiota instead of dead yeasts, or only accompanying the dead yeasts. So here, this taxon plot that came from Alpha Diversity. We see here on the left the panel A. This is comparing male to female fly gut microbiota composition. And then panel B, this is comparing flies with live yeasts versus flies with no yeast and its microbiota composition. We see in panel A that female and male flies, their microbiota composition is mostly the same. Sure, the peaks might differ in locations, but overall it appears that the patterns are very similar. However, in panel B, we see that flies with live yeasts have a greater abundance of this green color here. This is acetic acid bacteria, while flies without live yeast have a greater abundance of these tan and pink colors. These are enterobacteria and lactic acid bacteria. So in alpha diversity, it appears that there is an effect for the yeast. So studying beta diversity, this figure here on the left, this is the study of sex, the study of the effect fly sex has on the gut microbiota, and then this here is the study of whether live yeast presence has an effect on gut microbiota. So panels A, these are unweighted unifracts. Panels B, these are weighted unifracts. And panels C, these are Bray-Curtis tests. And for fly sex, none of them had a significant effect. And then same thing with yeast presence. Panels A, these are unweighted unifracts. Panels B, these are weighted unifracts. And panels C, these are Bray-Curtis tests. The unifracts, weighted and unweighted, had no significant effect. However, the weighted unifracts for the yeast did have a significant effect. So why is the Bray-Curtis saying there's a difference while the weighted unifract says there is no significant difference? I think it's because the weighted unifract takes how related the organisms are into account. So if there's a difference in the composition, but the organisms are closely related, then it will still say there is no significant difference. However, Bray-Curtis tests, they will not take relatedness into account. So if there's a difference in the composition, the microbiota, and even though the organisms can be very closely related, it will still see the difference and report that difference no matter the relationship of the organisms to each other. So that's why I believe the Bray-Curtis test does give a significant results versus the weighted unifract giving no significant results. And lastly, here is a PCOA plot on the right. This plot, the blue are flies with live yeast. The red are flies without live yeast. So squares are males, circles are females. And you can see that overall they're all pretty well mixed. Maybe there's a few like no yeasts over here. But like the main distances, these are created by the experiments that flies came from. So this is one experiment. This here is another experiment. This here was another experiment. And these a few here were another experiment. So overall, it appears that, and additionally, the ANCOM test revealed no significant difference at the family level or at taxon level 5. So, in conclusion, fly sex has no significant effect on microbiota composition. The presence of live yeast has a significant effect on microbiota composition at a species/genus level, but it does not have a significant effect at higher taxonomic orders. So, these are the conclusions for my study, and I hope that you enjoyed listening to this and learning about it.